Now something more convenient to your case is the application of Comet Assay in aquatic organisms. Is going to be given by invited lecturer Stoimir Kolev from the University of Belgrade.
Thank you, Marko. Our major interest in ecogenotoxicology is from the weight of the evidence aspect. I don't know if you're familiar with this concept. Actually, we are trying to prove by the evidence that we have some impact on the environment by pollution. And usually we are using few lines of evidence to prove it. So the first line that we are using is component-based methods. When you actually just go to environment, take water sample, and measure the concentration of xenobiotics in water. The second one is effect-based methods when you're actually doing in vitro tests in laboratory to prove that certain endpoint is triggered. Then we have in situ methods. I will talk about this today. So this is basically the response in aquatic organisms. And the last one is field-derived species inventories, which is meaning ecological status assessment. So basically what we want to do is to try to see if the xenobiotic molecule present in the environment can trigger the molecule or molecular response in your organism, and at the end that you can connect it with population response. So graphical abstract of our research would look like this. Actually, this is a graphical abstract of one of our papers. So basically, if you have some genotoxic driver in water, you can expect that there is uptake of this stressor in fish or mussels or any animal that you're using for your research. Then you can expect that genotoxic event will occur in fish tissues or any biota tissues. Then you should collect samples of the tissue. You should read analyses such as Comet Assay and try to prove that you actually have the effect in your organism. But in real life situation, it's quite more complicated than this. So when you're doing research in environment becomes your laboratory. So sometimes you need a big group of people just to collect your animals that you can use for your research. Sometimes you can go alone on the field to take your samples. Sometimes we have to set up improvised laboratory in the middle of the summer in days like this, and you have to keep all at some quality standards, which is very, very difficult. Sometimes we are doing research in the middle of the winter, and this is really horrible to perform, but sometimes it's really nice, and we have even time to take a swim if this is reference locations for our study. We are talking today about biomonitoring, about bioindicators, about biomarkers, but when we are talking about biomonitoring in these environmental studies, you can do it either by passive approach or active approach. The major difference is when you're doing passive approach, you're just doing assessment of the effects in animals which are collected at the sites of interest. So you just go, for instance, to river, you collect the animals which are already there, and you're running your experiments. The major issue that might occur is adaptation to pollution in these populations and also variability that you are detecting among the specimens. We will get to this later. And when you're doing active biomonitoring approach, you are actually exposing the animals from some sort of controlled populations to the site of interest, which is basically putting, for instance, fish in these kind of cages or mussels in these net cages, and then you expose them at the site where you're interested if you have genotoxic potential to be detected. Here, again, there are pros and cons. For instance, there is possible interference of translocation-related stress when you are exposing these animals to new sites. But how to get the quality data for Comet Assay from this kind of research? So basically, this is a roadmap with all the steps that you have to be careful when you're doing this kind of research. So when you're doing specimen collection, you have to take care of animal welfare. You have to avoid stress that you're causing to animals, and you have to have live animals to do experiments on. About tissue collection, again, be minded animal welfare on the first place. Again, try to avoid stressing your animal while you're taking the samples of tissue, and also think how to preserve your sample. Then, when you're doing preparation of suspension, you have to maintain cell viability. For Comet Assay, it is quite often that you have to have more than 70% of live cells in your sample. You have to know how to adjust the concentration to be optimal for Comet Assay, and again, you have to avoid causing stress to the cells in this case. For Comet Assay, you have to optimize your protocol, and you have to keep in mind historical controls so you can be sure that you're running it well. And at the end, mind the data and supporting parameters. DNA damage detected by Comet Assay is a biomarker which needs additional proof in means of supporting parameters that actually you are detecting the effect in the environment. So how one day in life of ecogenotoxicologist can look like? So when we have these kind of experiments in the field, usually we meet at 7 o'clock, we have morning coffee, we gear up, and we travel to location. It can last up to 3 hours if we are doing research in Serbia, so still it's morning. Then we should do sample collection. If you're doing active monitoring, that it's simple. You have cages, you just collect the animals from cages. But if you're doing passive monitoring, then, and if you are catching animals and if you're working with fish, this could be terrible and it can last for a few hours. And at the same time, DNA damage in us is progressing as we are doing the research. So when we have collected the specimens, we are doing dissection and tissue collection. We usually do it on field. Then we have to travel back to laboratory. We have to prepare the cell suspensions to check cell viability. It's already night, and then at 10 p.m. we have to start doing Comet Assay. We're all very tired, and it's becoming exhausting. The first phase of our research is study design. So you have to think how to select the location that you want to do study on. How to select the organism that you will use, and how to provide reference values. So this is the satellite image that I like to show quite often. We are in Serbia again. We don't have wastewater treatment plants. This is a section of the Sava River, also going through Croatia here. This whole section on the image is about 1 kilometer long and about 200 meters wide. It is situated downstream of town Obrenovac. It has about 50,000 people. So Obrenovac does not have a wastewater treatment plant. It is discharging wastewaters in this Kolubara River. It's a small tributary of Sava. And it's going all to Sava. It is indicated with yellow here artificially. It's not like this in real life. And also downstream of this confluence, you have the industrial area. They are treating their wastewaters, but still they are discharging it into Sava River. It is indicated with red color here. And if you're going further upstream, this green arrow points to the section of the river which actually has good water quality. It is used for recreation of the people for Obrenovac, and it is not unpolluted, but it is, as we call it, the best available reference site that we can get. So if you're doing active monitoring, and if you expose your cages here, you can be quite sure that when you're doing analysis in your organisms, that you're actually detecting the impact of the wastewaters of Obrenovac. If you expose cages here, then you can have the reference site for your research. Or if you go further downstream, then you know that you have detected the impact of these industrial waters as well. The similar situation is when we are doing passive monitoring using freshwater mussels. They are semi-sessile organisms, so if you collect them there, they are just living there for a certain period of time. We did some experiments of marking the animals and putting them back. If you come after 1 month, you can collect it again by diving the same animal with a mark. But if you're doing research for fish, for instance, on this site, we did it on freshwater bleak. It is small fish up to 10 centimeters long. It is Albunus albunus. According to the ichthyologist, if you collect fish here by small nets, there's a 70% chance that this fish spent more than 1 month in this area. So even if you collect it here, you can say that it probably spent only a few days in this polluted site. If you're going to do the experiment with fish big like this is northern pike. They are very migratory, so even if you collect it here, it just came probably that day to feed there, and tomorrow it will be somewhere else, and it came from who knows where, and we are not sure what we're actually detecting in these animals. Again, then you have to be careful how will you do interpretation of your results. Are they characterized as site-specific? So if it's mussels, we can say, okay, the response that we got is a site-specific, but if you're doing in fish, then we have to say it is stress-specific, and depending on the migratory potential of the fish, we can say how big is this section that we are actually characterizing. So when you decided on which animal you will do your experiment and how will you do it as active or passive biomonitoring, then you can actually start with obtaining data in ecogenotoxicology. If you're talking about, again, passive monitoring, we have to think about specimen collection, then the tissue collection, then how to transport the samples to laboratory, how to prepare suspension, and how to do Comet Assay. If we are working with freshwater mussels, it's quite easy. We can collect them with hand nets if they are there, or by diving. They usually inhabit depth up to 3 to 4 meters. So even with only with fins, you can collect them easily. If the site is not really polluted, then it's really not recommended to dive in there. And but if we are doing experiments with fish, then it gets more complicated. So you can do it by electrofishing. Electrofishing is very effective for smaller rivers, but for big rivers, not at all. It is time consuming. It is labor demanding because you have to bring additional experts on the field from field ichthyology, which actually know how to do it and how to set up current, so you can avoid current as additional stressors on your animals. Then you can do it by fishing nets. It is effective for large rivers as well. It is, again, time consuming because you have to wait till fish is caught in the net. It is labor demanding. Again, you have to have ichthyologists who know how to do it. And additional stressor could be time spent caught in net. Quite often we are getting support from local fishermen. They are very effective for large rivers. They know how to handle fish up on collection. Again, time spent in their net would be a problem, but quite often communication with these people, they're very specific, and it's really find a good partner from that field. This is actually fisherman Chaja. He was actually involved in more PhD studies at our university than some professors. He's really amazing guy. And finally, we have gill nets. They are very effective for smaller species. I already mentioned freshwater bleak. When you're doing with these species, you can be only a biologist to recognize it is very characteristic fish species, and it is time and labor friendly. They're usually caught in this net in less than half hour when you set them up. And additional stressor is untangling from the net because they are gill nets and they're quite often stuck in these nets. The problems that we face when we are collecting specimens is time frame needed for specimen collection. So as I said, it can be time consuming. You can pick the fish from one net and then you're waiting for fish from other net. And during that time, you have to keep the fish unstressed as much as you can. You have to take, again, care on animal welfare, and you have to use anesthetics. Quite often it is difficult to provide the equal number of specimens from each site, and quite often when we are writing papers, they asking us why do you have 10 fish at one site and only 7 another site, and you cannot explain that for other 3 fish we will probably need a few more hours to catch them. Also, sometimes it's impossible to provide equal number of specimens of both the gender. We had some studies, for instance, for northern pike, I showed it before. There should be equal distribution of the genders, but on this one site we caught 15 females and only 1 male. And tomorrow we did it again, and again we had this kind of gender distribution without any explanation. And a few days later, the fishermen sent me a photo that they caught 10 males and only 1 female. It is really a matter of luck. And also you have to mind 3R principle and to minimize the number of specimens that you're actually using in your research. And again, field work must not be harmful to natural populations. So you should not mind only 3R principle, but actually what are you trying to, how much harm are you causing to environment with your research? Regarding tissue collection, transport, and processing, we had already question from colleague here, and it depends how far are we from our laboratory. So if you are doing research on a freshwater mussels, they are very convenient for work. When you took them out from water, they just closed, and they closed a part of the water inside of them. And they are quite unstressed in this way. We never observed any effects on them when we are working, even after 1 or 2 hours after collection. If we are near the laboratory and if we can collect samples on field and transport them in cool boxes in next half hour to laboratory, we are doing that. If we are able to prepare Comet Assay slides directly on the site using there's like plates called gel bond. When you can actually prepare Comet Assay slides, they will not fall off. You put them in the lysis and transport it that way to laboratory, you can do it. But if you are not close to home, quite often we are using liquid nitrogen for cryopreservation of the tissue. So then you can you can carry out the study and do it later as you already suggested. Regarding the tissue that we are using for our research, when we are talking about fish, our primary choice is blood. Not the best choice, but the most optimal to work with. It is ready to use cell suspension. You have to just to adjust the cell concentration. After some experience, you can already know which dilution to prepare to use it directly for the Comet Assay without centrifugation or any other complicated equipment that you might need for field. And the most importantly, it can be cryopreserved well. On the other side, we use gills and liver, but in this case, you need to sacrifice the animals, and you need additional equipment to prepare cell suspension, which is difficult if you're doing on field. The protocols that we use so far are sensitive to cryopreservation. Amaya mentioned the protocol last time that we met that is actually working fine, but I still haven't had time to try it. I will. And these are good target tissues because gills are blood-water barrier, and liver is a place for transformation of the pollutants. When we are working on mussels, again, we are using most often hemolymph. It can be taken from the adductor mussel without killing the animal, as shown in image here. You can put the animal back to environment, and it will survive. It is again ready to use suspension. You can directly use it for Comet Assay, and also we are using gills and digestive gland. Again, you need to sacrifice the animals to collect this tissue. Again, you need additional equipment for preparation of your suspension, and the same reason they are good target tissues because gills are blood-water barrier, and again, in digestive gland, you have transformation of the pollutants. So finally, it is evening. We came to laboratory. We can start doing Comet Assay if we decided to do it all in one row. So here is an example how it actually looks like. For instance, if we have collected 5 fish, and we are doing research on 3 types of tissue, so it's 5 times 3. And you want to try multiple dilutions because we are not sure how many blood have you taken or how big piece of tissue has been homogenized for cell suspension. You have to try to do it in 3 dilution. So it is basically 9 Comet Assay gels, let's say it like this, or 9 Comet Assay slides. So if you have 5 animals, it is about 45 gels that you have to prepare. If you're doing standard Comet Assay, and that's why we are usually using this medium throughput Comet Assay, and on that way we can fit it all on 3 slides. As I said, you have to check the cell concentration. So if you have a lot of samples collected that day. It is difficult to do it on microscope one by one, so we usually prepare 3 dilutions and run it all together, knowing that at least one dilution will work nice for our study. Usually we are doing cell IVs overnight. We do it for 12 hours. It's, for us, it was shown that it is better than doing 1 to 2 hours only, especially if you have a lot of samples and if you're doing it in series. We are doing slide fixation in ethanol after neutralization. So we are making like libraries of slides and we are scoring them later. And we are using tail intensity as a measure of DNA damage. So this is just one example of for the study of passive monitoring that we did. We were involved in international expedition which was carried out on the Danube River. So we covered almost 3,000 kilometers, starting from Germany and it got up in the Black Sea. We managed to, the whole survey was performed on 68 sites, but we found mussels on 30 and something sites. We have collected 2 species of mussels and we did passive monitoring, just measuring the level of DNA damage in these animals. So here are the results. It's DNA damage measured by tail intensity. White bars are one species of mussel. The gray bars are other species. The white are for Unio, and the gray are for Synodonta boodeana. And at this point, I will only emphasize that the highest response in animals we have detected in this sector 6. This sector 6 is situated between Drava River here in Croatia up to Velika Morava River in Serbia. And this is this stretch which is affected by wastewater. So as you expected, we are getting the highest response in these animals collected there. For example of the active monitoring, this is the study that we performed in the Belgrade city. As I mentioned, Belgrade does not have wastewater treatment plant. It is just discharging directly into rivers. Now, on satellite images, you can see the point where wastewaters are discharged. This is actually how it looks like without using additional colors. So we use these sites we exposed mussels at these sites in these net cages. And you can see at these heavily polluted sites that we're getting the highest response in animals measured by Comet Assay, let's say after 14 days of exposure, while this like reference sites that was quite fine during this period of exposure.